start, okay? Good morning, everybody. For those of you who don't know me, my name is Vered Nochi, and I'm Executive Director of the Philadelphia Israel Chamber of Commerce. This is a delight to host you here today. We're anticipating a lot of people, but it seems like it takes time for all to join. We're all zoomed out kind of these days. A little bit about who I am and what I represent. Philadelphia Israel Chamber of Commerce is an independent nonprofit based in Philadelphia and serving the greater Philadelphia region, Pennsylvania, southern New Jersey, and Delaware, although we also have members that are here on the Zoom, even from northern New Jersey. We connect Israeli and local companies and academic institute, investors, and others for joint ventures. We try to help Israeli companies enter the market here, as well as you, if you Americans, local, to enter the Israeli market, whether through joint ventures, opening innovation labs, and forming different collaborations, agreements, participating in consortiums and more. We also represent a fund that is federal and Israeli. It's called U.S. Israel Binational Industrial R&D Foundation, or in short, it's BIRD. Annually, BIRD awards about 30 grants of up to $1 million each for U.S. and Israeli co-development projects. And we will help you connect with Israeli partners, or if you're Israeli with local partners, we will coach you and help you get the grants. Last year, we had 3 grants awarded here in our region. We also work with the Commonwealth of Pennsylvania, with the Commonwealth of New Jersey, and the state of Delaware to also work on other collaboration agreements. So if you'd like to pursue such relationships, please contact us. We're membership-based, so you're welcome to join us. Today, we'll have the pleasure and honor of recognizing the accomplishment of Temple University Maurice Kornberg School of Dentistry, led by the wonderful Dean Amid Ismail. And we will today honor him with our 2020 Life Sciences, Healthcare and Bio Award, and we will discuss it in more detail later. In addition, we have fantastic programming. We will have a panel discussion on the latest and greatest needs and solutions regarding COVID-19 testing technologies, rapid testing technologies that will enable us to open the market quickly. And we will present the panel pretty soon, as well as we will have 2 presentations of the award, two leaders to the dean. And most importantly, I guess, for all of you that are interested in testing kits, we will have Israeli companies presenting, and we will enable you to meet them face-to-face in breakout rooms. Thank you all for participating today. And without further ado, I'd like to introduce you to my dear friend, partner in crime in New York. His name is Inon Elroy. He represents basically the state of Israel as Minister of Economy for North America, overseeing the entire East Coast. Inon has been working for us, I would say, about 4 years now, in connecting companies, as well as on these projects and many others. And he will share with you some of the ways we can help you. And also, thank you so much, Inon and Josh Berliner, who work with me to make this program, as well as I'd like to thank Rafael Moog from the Israeli Export Institute, who made great efforts together with Josh and Inon to recruit the Israeli companies on this panel. So thank you so much. And without further ado, I'd like to introduce Inon Elroy, please.
Thank you, Vered. Good morning, everybody. It's great to be back in Philadelphia, even virtually. So welcome all the participants from the US, from Israel, and from elsewhere. I'm sure we're going to have a very interesting discussion. It is an honor for me to be here again with you, celebrating the Philadelphia Israel Chamber of Commerce and the incredible work achieved by your community in the name of peace, prosperity, security across the world. We have so many friends there, and we see the bonds are getting stronger between all the region and Israel. and we're very happy to be part of this. It is fitting that this year, when we are confined to our home states, the Chamber is honoring Dr. Ahmed Ismail. This is because despite being based in Philadelphia as Dean of Temple University Kornberg School of Dentistry, Dr. Ismail made an impact by bringing together dental experts from various cultural backgrounds and countries in order to bring dental care to patients of all stripes. His leadership has made waves well beyond the campus. It has brought 40 institutions from Israel, United States, Palestinian Authority together, in addition to other Arab dental and oral experts, to collaborate in advancing dental and oral care for the disabled at Shalva Center in Jerusalem. We salute Dr. Ismail for his service, and I thank you all for your continued support. Today, as Vered said, we're working together in order to bring together US and Israeli companies and trying to strengthen the excellent bonds which exist. Together with the Israeli Export Institute, and thank you, Rafael, we're pleased to present a group of companies from Israel that are developing the next generation of rapid COVID testing. These technologies will enable business and public spaces to reopen safely and responsibly so that we can celebrate the PICC's Gala together in person in 2021. We're looking forward for that and for all the, old things we used to do pre the pandemic. Until then, my office at the Israeli Economic Mission, together with the Israeli Export Institute, are working together with the PICC to support joint ventures with Israeli companies, products, and society. Our door is always open, and we'll be very happy to help you to identify relevant Israeli partners. So thank you again, and I'm looking forward for the program. Thanks.
Thank you so much. I will be remiss if I wouldn't thank the many friends of the Philadelphia Israel Chamber who helped us in actually making this program possible. I'd like to recognize our supporters, the Sattel Institute, our award sponsors, our platinum sponsors and members, our gold membership sponsors, our silvership membership sponsors, and others who contributed to support this program. Did I show this or not? I don't think I showed them. So here I am sharing this. I'm sorry. The Sattel Institute and our members. Thank you so much for the support. The PICC is a nonprofit supported by membership and sponsorships. Thank you so much. Now I have the pleasure to pass the torch to Julie Charleston. She's president and CEO of Premier Dental, which is a fantastic dental and oral company in the region, in Philadelphia, Greater Philadelphia region. And she will be leading our panel discussion. Without further ado, please, Julie.
Hello, thank you, Vered. Hello, everyone. It's great to be back for part 2 of our coronavirus innovation discussion. And last time, for those of you that were not with us, we were talking about the challenges of COVID testing in the healthcare industry overall, with a specific look at dental. So today, what we're going to be doing is focusing on needs and solutions. And the people that are going to be helping us to do that is our phenomenal panel, who I will turn this over for a brief introduction for them to introduce themselves each individually. And I will start with Dean Ismail for a quick introduction.
Good morning, everyone. Ahmed Ismail, I'm the Dean of Kornberg School of Dentistry at Temple University, also Laura Carnell Professor. I work with this topic every day, so I'm going to bring you my daily knowledge, experiential knowledge to share with you.
We're excited. Okay, Dr. Palmon, please tell us a little bit about yourself.
Hello to all. My name is Aaron Palmon. I'm the Dean of the Faculty of Dental Medicine, Hebrew University, Hadassah, Jerusalem. As Dean Ismail, we are working in this COVID-19 area in dentistry, so we are struggle with all these diagnostics to help us keep working. Thank you.
Thank you. The next member of our panel is David Kochman. David?
Thanks very much, Julie. Thank you to PICC, to all the participants, and to all the hosts. I am the Vice President of Corporate Affairs at Henry Schein, and along with some of my other colleagues who I believe are on this, have been working with our teams to help bring diagnostic testing outside of its traditional space. Henry Schein is the world's largest provider of product, services, equipment to office-based professionals, and we have quite a large footprint in the dental space. It's a delight and a joy to be here. Look forward to the panel. Thank you, Julie.
Thank you. And rounding out this fantastic panel is Liat Schneiderman. Liat, maybe you can tell us briefly a little bit about yourself and your work. And before you get started, just so you know, you're on mute. And possibly frozen. Okay, so while Liat is figuring that out, she's going to be a fantastic member. Let's start to talk about kind of the presentation that we're looking at. Lital, are you off of me? Are you able to hear me?
Yes, I'm able to hear you. Can you hear me okay?
Okay, good. You were frozen a little bit. Yes. So I'm going to ask you as the next member of our panel to just tell us, Lital, a little bit about your work and about yourself, and then we'll get into the presentations.
All right. So thank you, everyone. Pleasure to be here. My name is Lital Schneiderman. I'm the Director for Business Development at ARC, which is Shiva's innovation arm, and lead the innovation operation. And in the last 8 months, I overseed all our innovation efforts that were dedicated to the Corona effort. For us, this is a very challenging time, and we used all the methods, processes, infrastructure that we've built in the last 4 years to serve our Corona efforts in several fronts, not just diagnostics. And since today's panel is dedicated to diagnostic, I will just elaborate on that a little bit. And really, our approach is to be open to basically any method. So we're happy to collaborate with startups from all over the world using different technologies, from voice recognition testing application that basically can identify supposedly Corona based on voice, all the way through spectral technology, which we'll hear some of today, and also with the more of the conservative methods, the antigen, and of course the PCR. But the interesting effort is really to combine all, and sometimes build processes combining two methods and improve screening, allowing fast and safe basically testing.
Great, thank you, Lital. Thank you very much. So as you can all see, we certainly have an expert panel here with lots of diverse perspectives and insights. So with that, let's start by taking a look at what's happening globally. What are we seeing in other countries? What are they doing right? What are they not doing right in terms of data, in terms of diagnostics and testing? So with that, Dean Ismail, maybe you can shed some light on that for us, please.
Thank you very much, Julie. I want to take a global view, as Julie said, and also look at what things have worked and what things have not worked, and put testing in the context of a system. So we have a lot of experience, if we go back to the 19th and 18th century, when we didn't have vaccines, how we managed pandemics. So hygiene and public sanitation, poverty and housing, water supply system that's clean water, disposal of wastewater, and quarantine, which stands actually for 40 days, not 14 days. And that was usually used for ships and cities of towns when they experienced a large outbreak of an infectious disease. So at that time, they had no treatment, they had no vaccine, which is exactly in 21st century we have with COVID-19. So we're repeating the same thing that were done before, but now we have to look at to look at it from a different perspective. Masks is not a new invention. It has been in use, was used in the 1918 flu pandemic, and people were reminded, as this photos I got it today, is basically reminded if they're not wearing masks to wear masks. So everything we're doing today is not new, it's really a repetition of things we did in the past. However, today, because it's a different world than 100 years ago, we have a lot of communication, travel is easy, transmission of disease is different than it used to be. And I looked at different countries and what's the size of the population, the cases they have, and deaths. And you can see from this slide, when you compare Japan to the United States and Israel to the United States, that there are countries that have significantly lower number of cases and lower number of deaths. Taiwan, South Korea, Israel as well, though it's higher than Japan. So the question is why these countries have succeeded. Sorry about that, that slide moved. Now I have a frozen screen. Did I lose control? Okay, so I decided to look at there are some countries I didn't include because I cannot trust the data. Even some countries may not be the appropriate comparison. But I took Taiwan and South Korea, and I'm going to focus only on Taiwan. And the reason why is that we need to look at the fidelity and accuracy of the data. There are different age distributions. There are other factors in healthcare system and public health systems, and also political systems. Taiwan has a population of 24 million, 81 miles from mainland China, 850,000 live in China, 400,000 work in China, 227 million visitors from China to Taiwan. So they're really when the epidemic started, they are at the front door of the epidemic in China or pandemic in China. When that started, they are at the front door. So immediately when the WHO announced the problem, they on December 29 that the emergence of clusters of pneumonia of unknown origin, the Taiwanese officials started to board flights from China and screen patients for temperature and so on. The system has a National Health Command Center that was activated later on in January, which manages all actions, all decisions, all distributions, and reallocate resources in the country and also communicate. Something that not all successful models of countries that succeeded in managing the pandemic has or have. So this is something to pay attention to. In January 2020, Taiwan declared an emergency, activated the National Health Command Center, had the minister as the commander. He has a lot of powers. His approval rating now in the country is 80%. Masks were provided. They manufacture masks. That's another component, is manufacturing in the country is very important. They were selling them for 27 cents a piece. And each citizen have access to 50 masks. Use travel and health insurance record. When the extensive national health insurance database was matched with the travel database, and they were able to identify who traveled, what symptoms they have, and then screen them, call them and screen them. They didn't know what to screen for initially in January, so they screened for several viruses until the COVID-19 test was developed and manufactured in China. So that's another point about manufacturing. They have developed immediately in January, they had regional local testing centers, and all manufacture of PPE was in Taiwan. So that's a significant advantage for a country. Then they started to travel ban from China. So no one could travel from or to China. And then they added the other countries. The other thing that we cannot do, and many other countries do not, they introduced a $10,000 US fine, really, for anyone who violates the quarantine. And they have this is something that if you want quarantine, this is enforced quarantine. You cannot violate it. The other thing that's wonderful in the system is not just the testing, but they also have the contact tracing. So as I said, they have this big data that they access to. For example, they looked at everyone who went to a doctor who had flu-like symptoms, but didn't test positive for the flu. They called them back and they found 1 in 113 actually have COVID. So it's less than 1%, but they had COVID, they're positive for COVID-19, so they call them and start to quarantine them, of course. The QR sensing on phones, the QR code scanning on phones what helped in contact tracing. So Taiwan didn't go into a lockdown. They went into a system to manage the cases in a very extensive and deep way, which include testing. So they had commander communication, testing, PPE and prevention, big data analytics, case identifying and isolation very rapidly, and which is enforced, and that reduced the spread in Taiwan, which is very important. So to conclude, unified and empowered command center is very important. I think our Israeli colleagues could describe what they have done there because the IDF was involved. Rapid testing at designated centers. We need tests in different formats. We need tests for on-site clinics, hospitals, restaurants, businesses. Test at home would be ideal for people to test themselves at home, and testing of distal and proximal contacts of the cases. Sorry about that. Contact tracing, as I described it, monitored quarantine. Manufacturing is very important. We still run into significant problems in manufacturing in the United States. Everything is on back order. Mask, shield, gowns. I think this is a student at my dental school. Everyone wears a shield, and this is a level 3 mask. This is for outside the aerosol clinics. Air purifiers, disinfectant, we need technology. So that's the purpose of this session really, is to look at what is being developed in Israel in technology that we can bring it to the United States at a faster pace to implement it. Thank you very much.
Thank you very much, Dean. Well, I certainly can attest to, and you know this unfortunately firsthand, as does David, the disruptions that we're seeing in the supply chain and really what that's doing in terms of backing everything up. So now knowing kind of setting the groundwork overall, I'd like to move into what we're thinking in terms of ideal testing. So Dr. Palmon, I'd like to learn from you and have everyone else here as well, of course, what you've been seeing and thinking about in terms of ideal testing. You know, what components do they have? What characteristics do they have? So Dr. Palmon, if you could share that with us, please.
Okay, thank you. Thank you, Julie. Can I have my presentation now?
Oh, that's way above my pay grade. Somebody else is going to
No, it's not your job. It's not your job. It will take a minute.
Okay.
So my talk will focus mainly, no, I can't see my presentation. Okay. So my talk will focus on the goals and the parameters for ideal COVID-19 optimal test. So if we think about the goals of a potential test, it can answer 3 questions. One, which is clinical. A person wants to know if he has flu-like symptoms, if he is sick, and if he is sick with COVID-19, because this will decide upon the treatment he should get. Okay, so what we saw at the beginning was that. And later on, we thought about 2 other goals, which are connected with each other, epidemiological and economical. Both are intended to reduce the transmission within the population. If we want to stop COVID-19, we need to reduce transmission within the population. If we want to open the market, we need to identify people who may impose risk of transmission on us or on our customers. If we look at on dental clinic, I don't want my patient to infect me by a chance or by transmission infect my other patients within the clinic. Understanding these goals, we need to define what are the parameters, next slide please, that will define an optimal test in the dental field, but not only in the dental field, in all the services that the economy can supply to us. So first, the optimal test should be chairside, which means a person goes to the clinic, and when he enters the clinic, I want to know in this moment if this person is positive of COVID-19 or not. In the optimal case scenario, we don't want to send the samples and get the results after 2 or 3 days. And we want to do it by little medical staff or about not highly trained medical staff. A second definition is simple to use, that we want that our system will be easy to use, no lab work, no large equipment. Okay, because if you need lab work, then it will take a lot of time, or if you have large equipment within the dental field, this is the barrier. A third definition is that we want the procedure to take little time, up to 15 minutes. If we think about patient in a waiting room, you don't want to summon him 2 hours before. Okay, you want to summon him, finish your treatment, your dental assistant will give him the COVID-19 assay test, and okay, we will have the result. And fourth, we want it to be affordable. If it would be too much, if it would be expensive, then less people would be able to do this test, and it will stop the economy. So in the coming presentations of the Israeli companies, the companies try to cope with these definitions by several technologies. Some of the technologies that we will see are spectral-based technologies using oral rinse or aerosol breaths, and the others are using saliva. Okay, so we are trying to cope with this challenge either by new technologies or by taking samples that are easy to collect. So thank you for that time.
Thank you very much, Dr. Palmon. So with that, now kind of we're seeing a picture where you helped us to think about what ideal technologies would be, and coming up ahead with the Israeli entrepreneurs and their innovations, we're going to be looking at what these emerging technologies are. So what I'd like to do is bring us back to today. What exists in the market right now in terms of point of care? So David, maybe you can show us a little bit about what exists in terms of point of care, what tests are out there, where they are being administered, and then that can help to round the discussion out on the area of testing.
Terrific. Thanks very much, Julie. I'll move through this relatively quickly, and with the caveat that I am neither a dentist nor a physician, and so I really have to credit our team for pulling all of this together. At a high level here, right, there's really 3 ways that we can look at testing: 2 for the viral category and then the antibody category. In the looking for the virus or the presence of the virus itself, right, we have 2 ways that we can go about it. We can do a molecular test, which is actually looking for the viral RNA, or we can look for the protein coding, the antigen, that would indicate the presence of the virus. The key differentiators are obviously what you're looking for and then the stage at which they can be detected. And then the antibody test is looking for the immune response. This is a bit more detailed chart that we like, which helps to really illustrate the timing of when each of these can be detected. And so as you can see, the molecular test is going to be generally more precise because it's looking for the molecule, whereas on the antigen side, it's going to show up a little bit later, generally coextensive with symptoms coming on. And then as you see with the antibodies, you'll be able to detect those antibodies post-infection. Certainly if someone was asymptomatic, you'd be able to see them as well, likely. There's a lot we don't know yet about the antibodies, the specific titer level of antibodies that's required to confer an enduring immunity. And so I think that there's more we can talk about it. For present purposes, I'm going to focus on talking largely about the viral category, which is what we're looking at here, and specifically focusing on what's available in a point of care setting. So generally, these would be your rapid tests. I've included a PCR test on here just so you can get a fuller sense of what's available, and also because for most of these PCR tests, you're going to be collecting on site. So folks, even though it's not a point of care test where the results are given point of care, the collection would take place point of care. And for some people, that's enough to do. So again, this also kind of gives you a sense as we talk about what are some of the attributes or questions we're going to be looking at exploring in determining what is the right test to be doing in a various setting. So the PCR category, right, these are generally going to be your lab-based tests. These are the ones that require a big piece of machinery, have a high throughput. And then another type of molecular test. PCR is a molecular test. Another type of molecular tests are your nucleic acid amplification tests, or that also can be done using real-time LAMP technology. These are just, again, ways of heating up the molecules to be able to identify them. And then you have your rapid antigen tests that, again, are looking for the protein, the presence of the protein. Presently, and this is not I won't represent that this is a full slate of all the tests that are available, but this is a pretty good indication of tests that are available. And this is just from our own data, from what we are putting it out there. I think a key piece there to look at is that last column, which is the availability of these tests. Many of the manufacturers historically have looked at who has been buying, who already has an install base so that they can sell into, as opposed to trying to get into new markets where the manufacturers are wondering, will there be a consistent buy on the long term? If you think about it, most of these manufacturers operate on the razor model. They'll give you the razor for free because they want you to buy the blades. It's the same thing with these tests. They want you to continue buying different assays. So running COVID over and over and over will have an ephemeral life cycle, whereas people that will continue doing additional diagnostic testing, that's where a lot of folks are focusing. So again, this is just kind of an overview here for the rapid point of care molecular tests. There's 3 that we're presently seeing. You can see those up there, only one of which right now has good availability if you don't already have an install base. And then on the antigen side, similarly, BD Veritor just had a DPA put in. That's a Defense Production Act, which was a sequestration by the government to take a number of the instruments. We should hopefully be seeing additional availability coming down the line. And then the Binax now is a lateral flow assay, right? That's just one of these paper strip tests. The government, I think it was quite publicized that they went and took a large volume of those tests and have now, there is far less availability of those. We're not sure exactly when there will be greater availability, likely in the beginning of 2021. However, the comment I will make is that we do expect or hope to see some additional lateral flow assays come to the market. And then concurrently with that, there's some really exciting new technologies that we hope and expect will be able to be commercialized around that same time. And I think, Julie, that's probably a good segue to our other panelists.
Great, well, thank you. Well, you might not be a scientist, but you at least on it a little bit in terms of how you determine which test for which setting. Maybe you can elaborate on that a little bit more, and Dean Ismail as well, in terms of really not only which test for which setting, but also looking into who's going to be administering these tests in these given settings.
Sure, happy to do that. And maybe I'll just put this back up here if that's all right. Apologies. So, things we like to think about for different settings, and Ahmed, maybe I'll start just talking about the private practice space itself, and then and the occupational health space, and then can flip over to you for the clinic side. But 3 considerations, again, non-exclusive, but 3 things to be thinking about would be your throughput, the method of collection you're going to be using, and cost. Those are just things you want to factor in. So for throughput, how many tests are you going to be doing on in a given day, in a given week? Do you have a large group of people that are perhaps kind of coming in all at once and you need to test them all together? Are you doing it episodically where perhaps someone presents with some symptoms and you want to go and check? These are factors that you might think. In a dental setting itself, what we have been seeing from many of our customers, and just in a pilot that we are starting, is that people are doing very low throughput, somewhere on the order of 10 to 50 tests a month is what they anticipate. So you don't need a machine that's going to handle a whole lot. You just need something that you can use, and perhaps maybe you buy 2 cheap analyzers to run tests, because you don't need a large machine that's going to be able to do 10, 20, 30 at a time. The collection method also is really important. I think what we're finding, at least in the dental office setting, is that people are not really willing to do the full nasopharyngeal, the brain swab, but they are more comfortable doing an anterior nasal, essentially just right in the front of the nose swab, and especially would love to have something that is saliva-based. Something that patients can spit into. It's far, far easier, although I'll note with this, at least some of the saliva tests that are out there, there is a requirement that you fill to a certain level, and for younger children, that's become a challenge. For the occupational health space, though, something that you can do very quickly is a lot of saliva collections. And so for, maybe workplace right now, having a bunch of people come in, spin in a tube, and then taking those samples, sending them off to a lab is perhaps a better workflow for some workplaces rather than going and doing individual tests. And then finally, the cost. I'll flip down just to show you, if I can. Oops, sorry. The cost is definitely a factor here. With your molecular tests, as you'll notice, right, you're generally getting a higher sensitivity and a higher specificity, the ability to the positive predictive value and the negative predictive value, because you're actually looking for the presence of the viral RNA. On the antigen side, the costs are going to be far cheaper generally, and those lateral flow assays that you're seeing, those paper tests, are all antigen. The trade-off you're getting is on the performance, a little bit of the sensitivity and specificity. And what we're seeing some workflows that are being adapted to account for those costs by perhaps running a number of antigen tests as a first screening mechanism, and then for anyone who identifies as positive, going and running a more sensitive test, either a rapid point of care molecular test or sending it off to a lab. In the occupational health workspace, it may be enough for people to simply run an antigen-based test, a very cheap test, and then for anyone who presents as positive, send them off to a physician space to just be tested and to look further. The oral surgeons themselves have historically been doing a lot of diagnostic testing, although not in this category. What we're finding that they seem to like is using the higher sensitivity test because it's pre-surgery. Perhaps there's more dollars there to make up for it, or they just want to know, have that accuracy right up front as opposed to delay. So again, mindful that there's many considerations: what works, where to do the testing in your office, how many patients are you going to be seeing. The 3 considerations we generally look for would be your throughput, the method of collection and who's going to be collecting, where you have to do that, and the costs. And all of those factor into an overall testing strategy. Dean Ismail, I don't know if you have thoughts on the specifically on the clinic space where you have much more expertise.
Yeah, so it depends on what's the purpose of testing. But if I'm going to test a group of students or faculty or patients, and I want to get them into the clinic, I want to be sure that they are negative. I don't mind the false positives, because the false positive will be screened again or tested again by a PCR test. But I don't want to send someone to the clinic who I told them you're negative and they're positive, because they may be infecting 3-4 people, maybe 5 people. I mean, more than one person would be infected. The average is 2.5. So that's a very important consideration in selecting the test. And a large scale screening is a different story there. As again, what's the purpose of it? A lot of people do it, or institutions are doing, to feel good that they are isolating the positives. But the accuracy of the test will be a very important determinant. If we're using the test in a different way, which is if a person is positive in a unit and we go and test all the units, and some groups are doing group testing, they collect samples like saliva from or swabs from a larger group of 10 people, the idea here is I want to identify all positives very accurately. So the issue is a balance between accuracy of sensitivity and specificity of predictive value of these tests. Depends on the purpose of the testing.
That is very, very helpful and insightful, both of these pieces of information. So let's continue on with the management and the solutions. And with that, I'd like to turn it over to Lital. Lital, if you can tell us about your specific experiences with Sheba, what you're seeing in terms of the clinical and hospital settings, and how you're utilizing not just the innovation, but also the data and the technologies and the analysis around that.
Sure. So as of today, we're using the rapid antigen tests under clinical trials in Sheba as one of the screening methods for patients coming to the ER, and also to screen our team. And we find this to be safe and accurate, and serve as leading institute nationally to approve those tests nationwide. In terms of advanced technologies for ultra-fast technologies, we're mainly working with companies that use spectral imaging, two of which will present their solution later today. Specifically, VirusSite is a company formed in partnership of Sheba and NewSite. So this is the leading solution that we're looking to keep developing and testing. It's not a short journey, because sensitivity is an issue in any AI-based solution. Once you try different cohorts, ethnicities, geographic areas, the sensitivity becomes an issue. So you have to keep tune the algorithm and invest in developing the solution. We're also looking into other solutions based on identifying polyamines and chemicals. One is nanoscent, which is a test based on nose blow, and is looking to identify certain chemicals, and the other called Kidud, and looking for polyamines in the saliva. So the advantage in the ultra rapid test is definitely that they're less invasive, and none of them are nasopharynx, so it's easier to apply and possibly self-test yourself. But as for now, the antigen are basically more accurate, with the caveat that it takes 15 minutes and that you have to have a trained team taking those.
Great, thank you. So obviously, as with other industries, Israel continues to produce a lot of exciting and innovative technologies. So we've looked at the needs, we've reviewed the ideal solutions, we talked about what exists in terms of solutions and settings, and now we'd like to turn it over to learn about what the emerging technologies are, what the innovations are of the companies that are joining us here today in terms of their solutions for COVID testing. So Vered, I'm going to turn it over to you to introduce the panelists and the companies in that realm.
Thank you, Julie, first of all, for leading one wonderful panel discussion. You're very energetic, and you guys, all of you panelists were very informative and interesting. Thank you so much. I'm going to share screen and introduce our next, just a second. Just a second, not here. And I don't know why I'm not seeing it. Just a second. Here we go. I'd like to introduce you, Av Newman. Can you all see the screen?
No,
No?
no, we still don't see it.
Share screen. Can you see
Yes.
the screen now?
Yes, we can see it now.
Okay.
Yes.
It is my pleasure, sorry for the delay, to introduce Dr. Yoav Newman, and he represents Cell Diagnostic. Please, Yoav.
Thanks, Vered, and thank you all for joining us and for the opportunity to present our developments here. Can I just do it by myself, Vered? So Cell Diagnostics is developing a saliva-based diagnostic solutions. I don't think this panel needs too much explanation regarding the advantages of saliva as a diagnostic body fluid. I keep getting messages from the system. Should I share it from my computer?
What do you mean? Do you want me to move the slide, change the slide?
No. Okay. Saliva is convenient and easy to collect and allow home use devices that operate by the end users themselves. It is also enables non-invasive modes of collection, and in the right setting, it also allows rapid detection. Now you can change the slide. It was mentioned before, but our requirements from a rapid COVID-19 test would be a POC-oriented device to fit the emergency use authorization guideline. It should be easy to use, again, for self-testing without any additional equipment and other, even a power source. It should fit the chairside testing. It should be fast. We're aiming to obtain results within 15 minutes or less. And as again mentioned before, it should be affordable. Please go ahead. This is you should start the movie now. This is a clip taken from our saliva-based pregnancy test. It shares the same mode of operation as our COVID-19 test. The user inserts a collecting unit into the mouth until a sufficient amount of saliva is collected. Then it is inserted into an analytical unit, and then the saliva is squeezed over, in this case, a lateral flow assay. And like pregnancy test, in our COVID-19 test, in case of a positive or in case that this person is positive for COVID-19, 2 lines are visible. If he's negative, only 1 line is visible. Next slide, please. The components of the proposed device are 3. You have the piston in pink, which is a part of the collecting device. In gray, you have the analytical unit, which holds our technologies for saliva processing that enables the use of saliva as an analytical body fluid or diagnostic body fluid. These technologies are implemented on these two little sponges that you can see on the bottom. And in yellow, you have the diagnostic cartridge. Again, in this case, it's just a regular lateral flow assay, but it can be snapped out and be replaced with any other mode of collecting and to be stored, to be shipped for other usage. I think this is much easier collecting and handling method than the nasopharyngeal one. And I think this is a great advantages of saliva and our method compared to most of the other tests. Yes, Vered?
1 minute, I'm sorry.
Okay, can I go on?
Yes, please.
Okay, we are in ongoing clinical trial at the moment, and we've found that if we're trying to cope with what Professor Palmon said earlier regarding the certain purposes of diagnostic devices, if we're looking for people with the higher viral load, and we're aiming at those who were receiving PCR results with CT lows from 26 and lower, we're now sitting on 83% sensitivity and higher level of specificity. I think that since such test allows us to examine the same person day after day after day without having too much problems of compliance, I think it can be the start of a solution in regard to open public places like airport, universities, factories, and so on.
Thank you.
Next slide, please.
Our time is up, so please wrap it up and we will open for questions.
Okay, we're aiming for the point of care at the moment in terms of regulatory. Later on, it will be over the counter and a patient care setting device. And the last slide, please. I just wanted to mention that we are supported financially by the NIH RADAX program, the Israeli Innovation the Israeli Innovation Authority, and by private investments. Thanks.
Thank you so much. Yeah. Thank you. Panelists, would you like to address any questions, or if anybody from the audience would like to ask questions, please feel free to address them in the chat room. Any comments? Yes, Dean.
You're on mute.
I want to ask a question whether the sensitivity could be improved above 83%. Again, my point is that the negatives are very important. I want to be truly detect negative, COVID negative patients.
It divides into two answers, and I will try to be quick. The short one is yes. We are now optimizing the test, and we are seeing that our optimization is successful, and it becomes more and more sensitive. But I think I know that it may be not that popular, but I think that too much sensitivity is a little bit overrated in terms of the last two purposes of the test. If I can adopt Professor Palmon list of the purposes of the test, because if you try to find the super spreaders, if you try to find the ones with a higher viral load, I think that that if your sensitivity will be too high, you will miss everyone, because nobody will be allowed to go out. If you want to move on the market, if you want to let university work, if you want to fly around the world, I think too much of a sensitivity will not let anyone to go out. So it needs to be improved. I agree on that, but I don't think that a device like ours, like the one I proposed, is the one that is needed to compete with the one that hospitals and clinics are getting to with their PCR tests.
Any more questions or comments?
Just the
Can you
technical
tell us a
question.
little bit more? Oh, sorry, go ahead, Lital.
This would be short. Where do you currently running your trials you have?
We are a Jerusalem-based company, and we are running it in Hadassah Medical Center.
Okay, thank you so much. Dr.
I have a quick question
Eliezer,
on.
go ahead.
Hi, Vered.
Go ahead, Var.
Please, Julie. Okay.
I just
Dr.
had a quick
Eliezer,
regulatory. All right, Vered, take it over.
No, please, Julie.
Hi, this is Ina Belopolska. I have a quick question.
Yes, Ina, we can't
okay.
see you.
It's a little bit of a broader question, but with respect to the, it's wonderful to have, as you're talking about the sensitivity of the tests, do you think that some of the countries may be under-reporting, maybe whether, we don't want to get into political reasons, but possibly because of that, possibly because they don't have the right tests currently, and might that change?
The question should be addressed about the technology because we are now in the pitch session. If we can, we'll accommodate additional questions in general later. We have more companies to pitch, and I would like to try to keep the schedule so you can meet them in the breakout rooms.
Absolutely.
So I apologize, Ina.
No problem. Sorry about that. I'll move back.
But please keep it for later,
Okay.
for when we finish this session and move to the breaking room. Thank you so much, Dr. Newman. Dr. Eliezer, are you available for presenting right now?
Sure.
We don't see you.
Sorry, hold on.
I'd like to, and I can either share my presentation or you can try to share your copy.
I can share mine, that's okay.
Please do, if you can.
Okay.
And I'd like to introduce Dr. Rom Eliaz. He represents Centec Medical. Thank you.
Can you see my screen?
Yes.
Perfect. Okay, so, so Centec Medical is a company that specializing in breath and test diagnostic. It was established on the infrastructure of several successful experiments that over the last decade, including clinical trials, and we used to detect infectious diseases and cancers. And we were requested by the MOH, the Ministry of Health in Israel, to work on COVID-19. So we started to work on COVID-19 only 6 months ago. And our technology is basically based on the idea that every organism has a unique metabolic process that produces a particular signature. With the company technology, we can identify volatile organic compounds, or VOCs, in exhaled breath, and we can identify the direct compounds associated with the virus activity. Our idea is to basically open borders, and the idea of the company is going into a biological security. So, and doing that with digital tests using exhalation technology for rapid virus detection. And because we can have a metabolic fingerprint that is unique to each genetic situation, we can basically use it to identify in early stages of the infection, even patients that are asymptomatic. And the main idea we have is that, like the breathalyzer that is using today for analyzing alcoholic drivers, we can use with our technology early detection of asymptomatic patients, but also symptomatic with a greater than 90% accuracy. Now, this is really important to differentiate ourselves from all the other companies and technologies. All the technologies are based on PCR or comparing themselves to PCR. However, PCR is good, as you know, only for about 70% of symptomatic patients and maybe less than 40% for asymptomatic patients. We claim that today, with the technology that we have, we can, and the fingerprints, we can detect also asymptomatic patients, but in accuracy that is 90% as of today. And we can also update the virus mutation or new future viruses based on the fingerprints that we found. In the future, the idea is to basically use our biomarkers and upload it to a cloud database and then using it for the test. And as of today, we basically completed 2 clinical trials, one in Meir Medical Center, another one in the Army. We are currently conducting 2 more clinical trials in 2 more centers in Israel, in Soroka and in Shamir Hospital. And we are basically looking to have metabolic fingerprints in a few weeks. We already have a metabolic fingerprint, but to be able to take our technology for validation and basically go through all the regulatory procedure of FDA and the European regulatory agency. And we believe that in several months we will be out there. Our technology is going to be really rapid. In less than a few minutes in the first phase, and then less than a few seconds in the later phase. Also, our test is supposed to be cheap. It's going to be less in the several few dollars for each test. However, the machinery, as I said, we are looking into biological borders. The machine is going to be in the beginning something in between $10,000 of dollars for one machine. We can have it in a hospital. We can have it in a center. A patient can come and basically detect themselves, diagnose if they have it or not, and then use it for traveling or other. But basically, the machine is supposed to be on borders or in big centers where the patient can be patients or basically every person can be examined for a few in several minutes, can have an answer if it's asymptomatic, or if he has the disease.
Thank you so much. Time's up if you would like to say one more word. Otherwise, we'll open for questions. Okay. Okay. Thank you, Dr. Elias. Please stop the share and let's open for questions. Any of the panelists has any comments, or the audience? I think one of the things that we discussed when we did the rehearsal meetings, what's interesting is some of the units, it would be interesting to see the logistics of how it will be scaled and where it will be positioned in the hospitals or in the airport or in any of the other locations to really conduct the testing in the most efficient manner. As you mentioned, it might be in airport or in a hospital.
Vered, could I just ask a question?
Please introduce yourself first.
Mark Tikachinsky at Jefferson. I'm just curious, what's the basis of viral specificity? Because the volatiles are coming from presumably epithelial airway cells. So how do they, why do they create a different fingerprint for 2 different viruses?
Because it's not only the epithelial, that's a common thing that people think, but there is also changes in the metabolic changes. Even in antigenic orders. So it's not only the epithelial that is using it, but also if there is an antigen or some DNA changes, the metabolic changes in the cell will basically release different chemicals outside, and once you exhale it, you can identify these different organic chemicals.
Can I?
Please.
Could you comment about specificity, not only about sensitivity, with regard to your clinical trials?
What exactly do you want to know about the specificity?
Specificity, you know, if somebody is negative, maybe he will be positive in your trial or not? I mean, what is the
as of today, again, we are still in clinical trials, but the specificity that we had was 100%. None of the results that we showed as of today show if it's show the false negative or false positive. We got completely accuracy to the PCR. However, we did find some patients that we believe they are asymptomatic, which the PCR didn't reveal. And we found later on, because we are looking into it as well, that they did have some signs of the disease when they were tested again in a PCR about 2 weeks later.
Thank you.
Thank you so much, Dr. Elias. I would like to move to the next company. We have with us Michal Cooper Gzolovich, and she is with VirusSite. Michal, are you here?
Yeah, hello everyone. I'm just gonna
thank you for joining.
Presentation for a second. Yeah. So, do you see my screen?
Yes.
Yes.
Great. So, just a second. Sorry. Just want the connection to be better, so I don't have surprises as we had before. Vered, right? Yeah. Now
everybody's in Zoom land.
Yeah. Now I think it would be perfect. Just.
It's working.
Yes, we can see it.
Yeah, great. So thank you for the opportunity to present alongside innovative Israeli technologies. My name is Michal Cooper, and I'm Director of Strategic Partnerships at NewSight Imaging. And the company I'm going to present to you today is actually VirusSite Diagnostics. It's a JV company we just established and launched this week. Was very excited with the Sheba Medical Center. Lital from Sheba mentioned it a little bit before. So this is actually, I think, one of the first times we're presenting as VirusSite, so it's quite exciting. And today I'm going to talk about VirusSite as an AI software company for pathogens diagnosis based on spectral technology, which is NewSight spectral technology. Just a little bit of background. As I mentioned, VirusSite is a JV company of ARC, which is the innovation of Sheba, which is ranked #9 in the world, and it's the biggest in the Middle East. Lital was a little bit modest, but we are very grateful working and collaborating with top minds and with Sheba, and together we believe we can really bring the solution fast to the market. I'm going to show you how exciting it is in a second. And NewSight is a semiconductor company, which basically is our expertise is sensors, which for many markets, and even before, we have very unique spectral chips. And once the pandemic happened, we switched our abilities to take our technology for spectral diagnosis, which is in real time and in very accurate and affordable and portable way. And we found a way to implement it into virus detection as well. And I'm going to show you exactly how. So an overview of our solution is basically taking a device which works with light only. We can use both methods, also mouthwash or saline oral lens, or we can take the UTM of the swab test and put it inside our device. I'm going to show you how it looks like. You put it inside the device, you click, and then in a second you get your results. It sounds maybe science fiction, but it's not. It works, and we already proved over 95% in preclinical trials made in Sheba in the lab. And now we're doing overall 30 pilots around the world to prove it also in the field. Some of them are also with the currently happening right now with the Ministry of Health in Israel to validate the technology also in the field. And we also signed with great partners like ICTS Europe, which has 120 airports around the world. And we're also starting pilots worldwide to also put it in the field as in airports or any point of cares and etc. So this is quite exciting because our ability to have something portable and affordable placed everywhere using only mouthwash. You don't need those inconvenient or it's non-invasive as swabs. You can get a great accuracy, not at first maybe for screening, but we do believe from our results, I'm going to show some right now, we can even sometimes get more accurate than the PCR and have also some predictions of the city levels and viral load. And while that's what we're doing now, while we submit and get our fast FDA approval, we already submitted it with a large institute in the US and we hope to get it very soon. And what I wanted to say, the last thing about the slides, and I don't have a lot of time and I want to switch to results to show you how, is that we see our solution much more than just, a rapid test. We see this as a revolution for remote healthcare. So imagine you have something that can be updated just using firmware for the next mutation of virus or any kind of new virus. We actually tested other pathogens and established and validated our ability to distinguish between different viruses, tropical viruses as well, like dengue and chikungunya. It's less relevant to the topic here, but we actually seen that there's something, a new let's say, news we can bring to the healthcare world, and we're also doing and working parallel in other medical disciplines as well. So it's quite exciting. So how this magic works, the device itself, NewSight's device, SpectraLite, the one you see here, it's actually small. It's in the size, I don't have it now to show you, but it's in the size of the palm of my hand. It's like a very base spectrometer, but what is special about it, it's based on our special chip manufactured in Israel. It's only something we can do. We don't buy it off the shelf and manipulate it. It's something very innovative that only we can do, and we developed it already for about 4 years, 4.5 years since NewSight came to life, and it's backed out, powered by machine learning and AI. And basically what we are able to do is detect the reaction of the patient to the virus. So basically we do not detect the virus itself. We can detect a spectral signature of the reaction to the virus. And we already validated that it's very accurate, and we can distinguish between different reactions for different viruses. We have done that. So I can show you some results. I don't have much time, but I'm just going to show you quick results. This is something we can show you because this is very premature. These results were the first we did in the preclinical trials in Sheba. Things we did after that I cannot show in this forum, but this is already good enough to show you that even in the beginning, we had already, if we're talking about specificity and sensitivity, you can see you have some of the data here. Although those specific tests were down at that time at about hundreds of patients, and since then, of course, we moved on to much more, but you can see a clear classification between positive and negative patients for COVID using mouthwash, basically straightforward saline that you can buy at any pharmacy, which is neutral. And this is another interesting thing, and that's all I can show you for now because I don't have much time. Time
is up, so please wrap up. Thank you.
Yeah, I know. That's why. We had an ability to predict the city levels and viral load of patients. So basically, we put it on a regression curve, and you can see in a very amazing R-square. So for now, we're confident that we can put them in between medium, low, and high, but eventually, since it's an AI learning engine, once we get more samples and we have deep learning, we are really confident we are gonna even be able to predict very accurate numbers, and maybe then to even replace PCR tests with the accuracy. So this just shows, I'm gonna wrap it up and just show you the mechanism of how it works. This is the mouthwash, this is the we can do the same for after the swab, we can use the diluted liquid of the UTM and also put it inside the spectrolite, as you see here, in a small disposable cuvette. And then you can press click. You have it available on PC or in our app, which will be available very soon. And you get positive or negative, and everything works using digital blockchain tokens. So everywhere in the world, you can get tested if it's in point of care, and eventually, we really hope that in the near future, even at home. So thank you very much. And I actually left Rotem Sorek information here. She will be also the one who will be in the meeting area later. She is of the Director of Business Development at Fireside. Unfortunately, she couldn't do the presentation now, but she will be later on there to answer all your questions and let you know more information. Thank you, and sorry for the extra 2 minutes.
One more question, Michal. They ask, how long does the test take? Like, how long does it take to get results?
1 second. Since we can obtain, we can obtain thousands. So basically, our chip works around thousands, 10,000 frames per second. But for this specific application, we take thousands of spectra in 1 second, and then we can generate enough data to create the spectral profiling. And since everything works very fast, we can very fast give a result if the spectral signature is matching to the profile of the sick or profile of the healthy. Okay. Just in a nutshell. Yeah, sorry, it's so simple. I don't have time.
Another person is asking, when you say accuracy, do you mean positive predictive value?
So accuracy is basically already combined after. So when I say 95% is something that we were really careful not to just throw numbers, and we have a document from the hospital that is generating basically validating this accuracy, which is already contained inside the specificity and sensitivity. We didn't have that much problem with false negative in the beginning. In the first place, we did have to deal with some false positives, but once we made the model better of the AI, we overcame that, and we see now in the results we're having in this pilots right now that we definitely can generate a very high accuracy in terms of screening, and hopefully in the future also replacing PCR.
Thank you so much. I would like to thank the 3 companies. There is another company that was listed in our agenda. They will not be able to participate. We have actually a very innovative company that joined us last minute. But at this moment, I'd like to proceed to the presentation sessions, and because this is exactly why we actually gathered here for the first place. And then if we have time, we will accommodate another company that actually is conducting tests with voice recognition. Literally, you talk to it and it recognizes if you have COVID or other things. So this is quite crazy, and we'd like to accommodate it. But first thing first, please stick around. We have the award presentation, and then we will have breakout rooms with the companies, so you'll be able to address more questions and actually talk to them directly. I'd like to, first of all, thank the 3 companies, Salignostics, Centech Medical, and VirusSite, a terrific contribution to us all. Thank you so much. And with that, I would like to, it's very exciting to introduce our former board member, as well as the current advisory board member, Rabbi Gary Charlstein, who is a chairman of Premier Dental, who nominated Dean Amid Ismail for this prestigious award. Please, I'd like to introduce you, Rabbi Charlstein.
Thank you very much, Vered. Thank you. You know, as a member of the Philadelphia Israel Chamber of Commerce, I have long been involved in these award presentations and seen some wonderful people presented for some terrific accomplishments. I therefore hopped up and said, hey, I think I have a wonderful candidate for just this award, because what this award signifies is bringing together of the Philadelphia area and Israel. Well, Dean Ismail has been with us here in Philadelphia since October of 2008, coming to us from the University of Michigan, having graduated from the University of Baghdad, and immediately went to work. we at Premier had a relationship already with Temple. And I'm not talking about the fact that my father of blessed memory was an alum of Temple, and my son went to Temple, and my daughter, who you've heard from, Julie, got her MBA there. But we were supplying with Temple, and they were involved with us with the various projects. But Temple, while it was an excellent school, was coasting for a while. But when Dean Ismail came in, he went right to work and immediately upgraded everything and changed it physically and in terms of the outreach to the community and the programs. It was a tremendous spark to the Kornberg School of Dentistry. And then we found a true friend of Israel, a true friend of peace, a true friend of inclusiveness. Hopefully, we've all already been impressed with Dean Ismail's research and scholarship. But to tell you a little bit about his Menschlichkeit, about his personality of an individual of great character who is so open to bringing together a variety of people to approach these significant problems, be they of providing dental care for the North Philadelphia community or bringing peace between Palestinians and Israelis. It was with Dean Ismail in Jerusalem, together with Adam Stabholz, his friend, then the dean, and the late, great Dr. D. Walter Cohen. We went to see the Al-Quds Dental School run in the Palestinian territory, and their dean, Musa Boujali, who could bring together scholars from their faculty with faculty from the Hebrew University. And beyond that, Dean Ismail partnered by bringing Israeli and Palestinian students here to Philadelphia to shadow students at Temple University. And we had the wonderful opportunity to meet these four and then another group of students and take them to some dental offices here in Philadelphia to share some ideas about practices. Very interesting, because there was a father and daughter combination here, and this young lady was working in her father's practice in Palestine. So it was quite an interesting exchange. And this is what Dean Ismail is all about: building bridges for peace, taking the great advances in dentistry and applying them to bringing people together, not only patients and doctors, but of course, those people that are concerned with finding solutions, be they health solutions or political solutions. And he does that with a force of personality and a sense of timing and of openness to new ideas. And that's indeed what this whole council is all about: bringing new ideas from Israel to the greater Philadelphia area, bringing ideas from the Philadelphia area to Israel and the greater Middle East. So whether it be with Turkey or Pakistan or Taiwan, we have seen tremendous openings that the Dean Ismail has afforded us. And so I would encourage all of you to get to know Dean Ismail. Come visit the Kornberg School. Involved him in some of your projects, you will not be disappointed, because here is a man of great integrity and great knowledge. I thank you, Vered, for this opportunity, and Dean Ismail, congratulations on this wonderful, well-deserved award.
Thank you.
Thank you.
Now I would like to introduce Rabbi Samuel Kalman, and he heads the center, which is called Shalva. It's a medical center in Israel for disabled, and he will be sharing with us his perspective on this award. Please join us. You're muted. I'll unmute you. Rabbi Kalman, can you unmute yourself? I'm trying to unmute you.
Unmuted.
Yes.
Great.
Thank you so much. My dear friends, thank you, Rabbi Charleston, and thank you to those whom you've recognized, a number of whom I have the privilege of being affiliated with. I'm humbled to have been asked to co-present this illustrious PICC award to the Temple University School of Dentistry and its illustrious dean, Hamid Ismail. Rabbi Charleston, you have shared a number of Dean Ismail's professional and menschlich accomplishments. I would like to share additional personal insights into the unique human being and dear friend that I have come to know and so admire. Allow me to give you some brief background. Born and raised in Vancouver, Canada, a child of the 60s, at the age of 18 while en route to university studies in Europe, I visited Israel and wound up staying, marrying my wife Malki, and studying to become a rabbi. Our second child, Yossi, was injured at the age of 11 months and became blind, deaf, and hyperactive. A well-meaning visitor suggested to Malki that she institutionalize Yossi. That night, Malki cried and said to God that she will never take Yossi out of the house, but if he ever decides to help our Yossi, she will dedicate her life to helping other mothers, she said, who are crying along with her with their children with disabilities. At 8 years of age, Yossi had an amazing breakthrough to communication when a teacher in the deaf school put one hand on the table, on the other hand, she spelled the Hebrew word for table. And after days and days, Yossi lit up with a smile, and she had the smarts to know he had just had his Helen Keller breakthrough. He went on to learn speech, and today he's a fully functioning individual in so many ways at the age of 42. At the time, Malki sat me down and said, it's payback time. We established an organization named Shalva, which means peace of mind, in Jerusalem in 1990, with 5 children in one rented garden apartment. What can I say? It seems that God has a great sense of humor. Today, 30 years later, the Shalva National Center in Jerusalem is one of the largest and most advanced facilities for those with disabilities, and provides programs and services for 2,000 people every day. The programs operate around the clock, serving the full cycle of life from infancy through adulthood for the entire social spectrum of Israeli society. I had the good fortune to meet Amid a number of years ago when he attended a dental conference in Israel and took the time to visit Shalva. I was overwhelmed by his love for the children, his warmth, and his enthusiasm. He met my son Yossi and immediately told me to order a 1-year subscription for him to The Economist, since it would stimulate his brilliant mind. I did, and Yossi enjoyed it immensely. Who else would have thought of that? Late that night, I received an email from Amid with a moving and brilliant poem that he wrote describing the emotions and the impact his visit had on him. Excuse me. In 30 years, no one else has sent me a post-visit poem. When I created an oral health program at Shalva, it was Amid who named it Oral Health for Everyone, because the 4 first letters of the Hebrew words mean ohev, or love. And it was Amid, together with Temple, who decided to bring contingents of dental students to Shalva to learn about oral health. But more importantly, the goal was to gain the experience of interacting firsthand with youngsters with disabilities to enable these aspiring dentists to better serve this population in the future. Amid refers to me as his rabbi, and I refer to Amid as my imam. So as Amid's rabbi, allow me to share that the weekly Bible portion read this week in synagogues the world over is the story of Noah and the flood. How meaningful this timing is when the stormy seas of our own time envelop us. Amid, in the spirit of Noah, is the ultimate builder of not one, but many arks, always seeking to protect and nurture those needing his help. He's forever focused on bringing divergent forces together, inclusion and never exclusion or divisiveness, always focusing on abilities and never on disabilities. My friends, I sincerely believe that building such arcs is the calling of our time, and no one I know does this better than Amid. Congratulations, Amid, on this great honor, and may we take your lead and continue building protective arcs for all who need it, each of us in our respective fields, and may we contribute to healing our wounded society. Congratulations.
I'd like to invite all of you to unmute yourself and join me in cheering for Dean Ismail.
Thank you. Thank you very much.
Yay.
Thank you.
Congratulations.
Thank you very much. And this is the award here that I have in my office. It's very, so Verd, if I may say a few words, I want to thank you first, the Philadelphia Israeli Chamber of Commerce. I'd like to thank Rabbi Charlstein for nominating me. It's a family that has connections with Temple, and one of the first groups that I visited in Philadelphia was the Charlstein family, Julie and Charles, and talked to them about how Temple could work with them. And then one day, I made a mistake. I left my coat and I was going to support the Alpha Omega meeting and the presidency of Dean Stables, who, but at that time, many people canceled their trips to Israel, and I was feeling bad about that in a meeting, and I decided to buy a ticket and fly to Israel to be with Dean Stables. I dropped my jacket in the airport in the car and decided, well, Israel may be warm. Now, I know Israel is not warm in December, and that was a very cold December. So I ended up going to meet Rabbi Samuels, not knowing what I was going to. It was an opportunity to sit in a taxi with Steve Katz and Alan Finkelstein, and it is warmer there in the taxi. Well, his heart, I discovered, his heart is like the sun. He is a person who has great warmth, and his devotion to serving the children with disabilities and adults, and his vision in concrete. We saw 8 floors of concrete, and now we're seeing, I visited 3 times, and it's a wonderful institution. It's one of a kind in the world. The Rabbi Samuels is my rabbi because he connects with me to my heart. And when I visited with Yossi, it was a friendship as if he's my son, and Rabbi is my brother. Then I thought about it, what a powerful message, what a powerful force that children with disabilities could bring in linking 2 groups of people, the Israelis and Palestinians and US students. So we started this project with Shalva, and its brand new idea is to bring students from the United States to meet their counterparts in both Israel and Palestine at Shalva. And what happened is that our students, we now send about 20 students, have been transformed. They come as different people when they spend a week at Shalva. And that is really the importance of bridge building. I think the Bridge to Peace is not my project. It's a project started way before I get engaged. The credit goes to many people, including Dean Sables and Dean Bajali. I was in Israel in 2012 or 13 in a meeting, and I saw that the Palestinian students and faculty were separate in a meeting about the Bridge to Peace. So I said, that doesn't make sense. So I started the Temple as another arm of that bridge to bring them to Philadelphia. And the great transformation is that to see young people from Israel and young people from Palestinian Authority started to understand each other, start to talk to each other as human beings, having shared values, and they became friends afterwards. And that is really the power of building bridges and understanding with each other. And I think I learned a lot that it's important to go see the other people on the other side who we don't understand them, is to understand them and see how they live, because there's a commonality in the human experience. So this is really a wonderful gift. I will cherish it all my life, and I will continue to work with Rabbi Samuels, Kalman Samuels, and with the Premier Group and with you, Verd, Nochi, so that we can really, and I can pronounce Nochi, by the way, so we can build new things in Philadelphia and also in Israel and Palestinian groups as well. Thank you very much.
Thank you so much. And on behalf of, I think our president may still be here, Matt Fingerman, as well as my vice president, Todd Abrams with Temple, I'd like to thank you for accepting the award. I'd like to thank you for playing with us so quickly and so amazingly to regroup after we couldn't host the gala and create two wonderful events featuring technologies that can help all of you go back to business very quickly. Also, the dean is very enthusiastic to try to pilot all sorts of technologies at Temple. Temple is a center of, among other medical institutes that are experts on dental care. We encourage all of you here to reach out to us and collaborate with the dean and with the PICC to pilot, to test, to bring to market technologies that are either in oral and dental care or any other that can help us go back to business. I'd like to, first of all, thank the Dean, Dean Ismail. I'd like to thank our economic attaché, Nona Roy, for joining us. I'd like to thank Julie Charleston, President and CEO of Premier Dental, for moderating, and our wonderful panel, Professor Ron Parmon, Lital Schneiderman from Sheba, which, by the way, Sheba is doing a lot of collaborations here with Jefferson and others, and it's only the tip of the iceberg of what we could do. We could do so much more, and thank you for joining us, as well as David Kochman with Henry Schein. We're here for you, all of the panelists and other. We're here for you to support your joint ventures, and we will now proceed for breakout rooms. You can contact me following if you need connections to any of the participants. We had about 153 people that joined, or at least registered. If they couldn't make it, I'll be glad to make introductions. Thank you so much. And now it will take a minute for Katrina to split us to room. Thank you so much.